My name is Kirtana Rajagopal, and today we will be talking about the adaptive regulation in the uptake process of TPP in the colon. But first, let's start with the basics, the digestive system. All living organisms must get nutrients from the environment to sustain life. These nutrients are used as raw materials for synthesizing essential compounds that are also broken down to provide energy or fuel that cells need to continue functioning. The digestive system consists of a muscular tube, or the digestive tract. It begins at the oral cavity and continues through the pharynx, esophagus, stomach, small intestine, and large intestine, or colon, and ends at the anus. But today we will be focusing on the colon. Some of the functions of the colon include conservation of water and electrolytes through reabsorption. It removes water, salt, and some nutrients, forming stool, which leads us to the second function, storage and controlled evacuation of the stool. The third function is that the colon acts as an important metabolic organ, thus influencing the overall metabolism. Additionally, bacteria inhabit the large intestine and contribute to the microbiome. Most of the gut bacteria live in the large intestine where they break down nutrients not absorbed in the small intestine and produce vitamins. The vitamin that we will be exploring further is vitamin B1, also known as thiamine. Specifically, we'll focus on the main active form of B1, known as thiamine pyrophosphate, or TPP. It is an essential cofactor for oxidative metabolism and reduces cellular oxidative stress by bridging the glycolytic and pentose phosphate pathways. TPP acts as a cofactor for several enzymes involved in the energy metabolism, including pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase. The vitamin also plays an important role in maintaining normal mitochondrial function and structure, since a deficiency in B1 causes oxidative metabolism impairment, resulting in the mitochondrial swelling and disorganization of the cristae, leading to cell mortality. It is important to note that the deficiency of this micronutrient at the cellular level would lead to impairment in the normal energy metabolism, oxidative stress, and mitochondrial abnormalities. Humans are unable to synthesize thiamine de novo. Therefore, we need to obtain this essential vitamin through various sources via intestinal absorption. Two sources of thiamine include a bacterial source and a dietary source. The bacterial source refers to the bacteria that line the colon, as mentioned before. They are capable of generating vitamin B1, specifically a free form of thiamine. The dietary thiamine exists as TPP, which is hydrolyzed to a free thiamine by small intestinal phosphatases before absorption. Previous studies have shown that both forms of the vitamin are absorbed in the large intestine, which, with absorption of the free thiamine being mediated by the SLC1982 and SLC1983 systems, while TPP is mediated by the SLC44A4 system, also known as TPP transporter or simply as TPPT. This transport will be the main focus of my paper. So the SLC44A4 gene produces a multi-transmembrane protein that is highly specific and has a high affinity for TPP. Thus, it mediates the cofactor levels by carrier-mediated process for TPP uptake. Another focus of my paper is adaptive regulation, which is regulation based on the extracellular environment or concentration of a substrate mediated at the transcriptional level. For example, results from the previous study show human intestine cells that are maintained in a TPP over supplemented growth medium leads to a specific and significant downregulation of the initial rate of TPP uptake by these cells. So, the adaptive regulation of TPP uptake process by extracellular substrate level seems to be mediated at the transcriptional level. Additionally, expression of the TPPT system has only been in the large intestine with negligible expression in the small intestine. Researchers have also cloned the promoter of the SLC44A4 gene and identified important roles for the transcription level factors ETS, ELF3, and CREB1 in driving its basal activity. This takes us to my paper, Molecular Mechanisms Involved in the Adaptive Regulation of Colonic Thiamine Pyrophosphate Uptake Process by Anandam et al., whose primary focus is to elucidate the molecular mechanism that mediates the adaptive regulation process. The experimental design for this experiment includes 6 steps. Researchers used a human-derived non-transformed colonic epithelial cell line, NCM460, with which they performed uptake assays where the cells were maintained for a short 2-day term and a long 9-day term in the presence and absence of unlabeled TPP. This was done to measure the custom-made TPP with hydrogen isotope uptake, in both short and long term. A Western blot is done to determine whether the reduction in TPP uptake is mediated via a decrease in the level expression of SLC44A4 protein. Colloids were then generated to use later in the RT-PCR, followed by a transfection of the SLC44A4 promoter construct and luciferase, followed by reporter gene assay and a chip assay. Based on the results, it was seen that there was a significantly lower rate of hydrogen isotope-attached TPP uptake by cells maintained in the presence of TPP compared to cells maintained in the absence, leading to a Western blot assay to determine whether the reduction in TPP uptake is mediated via a decrease in the level of expression of the SLC44A4 protein. As seen in panel B, there is a significantly lower level of expression of the SLC44A4 protein in the membrane fraction of the cells maintained for a short term in the presence of TPP was observed. Results with the long-term exposure to TPP as seen in panel D shows that there is a significant reduction in the level of expression of the SLC44A4 protein in the membrane fraction. Both Figure 1 and Figure 2 suggest that different mechanisms are involved in the adaptive regulation of the colonic TPP uptake process upon short and long-term exposure to extracellular TPP. Based on the transfection procedure, the results showed a significantly lower SLC44A4 promoter activity in cells maintained in the presence of TPP compared to those maintained in the absence in panel A. Interestingly, activity of the SLC44A4 promoter was similar in cells maintained in the presence and absence of TPP for the shorter period. Collectively, all results show that the adaptive regulation occurs following short-term exposure of colonic cells to TPP appears to be mediated via a post-translational mechanism, while that which occurs following long-term exposure involves a transcriptional mechanism affecting the SLC44A4 gene. Maintaining NCM460 cells in the culture medium containing TPP for a short period was not associated with changes in level of expression of the SLC44A4 protein in total cell homogenate. Rather, there was a decrease in the level of expression of the transport protein in the membrane fraction of these cells. On the other hand, maintaining NCMs for 60 cells in the presence of TPP for a long period was associated with a significant decrease in the level of expression of the SLC44A4 proteins as well as in the level of expression of its mRNA. Thus, it can be deducted that there is a difference of adaptive regulation between short and long term. Additionally, the significance is that the adaptive regulation in colonic TPP uptake process by prevailing extracellular substrate level is mediated via a post-translational mechanism. For class discussion, I would like to ask, what are other examples of adaptive regulation seen in the body? What would be the substrate that is mediated? Additionally, alcoholism could lead to thiamine deficiency. How and why does this happen based on your knowledge of TPP mechanism? Thank you.